Hello, my name is Beatriz Rosa de Azevedo, and I'll present you our study in silico drug repositioning for malaria from transcriptomics data. Malaria is one of the most important parasitic diseases in the world. It's caused by protozoal parasites of the genus Plasmodium, which are transmitted to humans by Anopheles mosquitoes. In 2019, there were more than 200 million cases and 400,000 deaths by malaria in the world. But until now, there aren't approved vaccines to this disease. This parasite has a complex life cycle with many stages. Combination treatments with different classes of antimalarials are widely used to reach more stages and hit different targets, because there are already resistant strains to most drugs separately. Therefore, it is of great interest searching new targets featured in multiple stages. Transcriptomics can help in this search. It is the study of gene expression and analyzes specific cellular moments. This way, it may be used to search therapeutic targets considering different development stages. For Plasmodium, there are online databases with lots of information on each stage's expression profile, making this study possible. The finding of new targets allows searching for new drugs. The process of drug discovery and development is long and expensive. An alternative is drug repositioning, which discovers new therapeutic uses for already approved drugs. This reduces time, costs, and the risk of the process. Considering this context, this study aimed repositioning drugs as antimalarials through potential therapeutic targets found in the transcriptome of Plasmodium falciparum and vivax. First, genes expressed in multiple stages were selected in the PlasmoDB platform for Plasmodium falciparum and vivax. From phenotypic essentiality data were identified the essential genes. From the falciparum genes were obtained the resulting amino acid sequences, and then these were searched in two therapeutic target databases. Converging the data, homologous targets and associated drugs were found. These data were checked by sequence pairwise alignment in a BLAST P2 at NCBI. The associated drugs were then submitted to a similar compound search using the bioactivity profile in the chemical checker platform. With this technique, it was possible to expand 20 times the compound set under study. At last, the drugs and similar compounds were evaluated by a virtual screening with QSAR models generated by LabMol. These models predict activity against sensible and resistant strains of Plasmodium falciparum in blood stage. Among the similar compounds, 94 were predicted as active and 11 drugs. In conclusion, this study used expression, drug targets, and available drugs data to make an integrated bioinformatics in silico repositioning strategy and found new antimalarial candidates. It was possible to find simultaneously expressed genes in various malaria stages, among them many essential and homologous to known drug targets. Associated with these targets were many drugs and a considerable number of similar compounds. At last, QSAR models predicted some of these drugs and similar compounds as active in blood stage malaria. Finally, further studies to experimentally evaluate the selected compounds in cellular assays against Plasmodium falciparum will be conducted. We thank the institutions who participated in this project and provided research licenses. Thank you for watching.